All right, getting started. So we're going to give it a couple more minutes for some more people to file in, and then I will share my screen and we'll start playing a Kahoot. Again, just to see what you guys know and what you may need to focus on a little bit more. And then in the second Zoom session, which I will also record, we'll go over any questions that you guys have. So how are we feeling so far about the exam? You can type in the chat or unmute. Don't want to do it. Unfortunately, nervous, but okay. Nervous. All right. Well, hopefully that this review today will help alleviate some of those nerves. Yeah. Showing up is half the battle. Okay. I think we're going to give it to like 3:05 just to get started, because Zoom will kick me off after 40 minutes. And then we will start another one. But I would rather finish the Kahoot in one so we don't have to bring it over into the next one as well when everyone's signed back in, and that just gets confusing. All right, so one more minute, we'll see what other people file in, and I will start pulling up this Kahoot. Okay. All right, it is 3:05. I'm going to share my screen, go to kahoot.it, and enter the game pin if you would like to play. I would recommend these are, some of these questions may be on your exam, or similar questions may be on your exam, so always good to do practice questions. And I will also be sending this Kahoot out with the recordings. If you want to practice a bit more, you can go through this again. You all have little icons. I wonder if the icons are from the theme I made, or because I've never seen those before. They're fun. Put this up top. Okay. I'm going to give it a couple more minutes for people to get in. But all right, is everyone who wants to be in? Yeah. Okay. 5 players, and then if you want to enter later, you can feel free. Or if you just want to watch, that is also there. All right, I'm going to start it now. All right, what's a retrovirus? Yes, that is correct. A virus whose RNA is transcribed into DNA in the host cell. Be careful with that one. Retroviruses are RNA viruses. They're not DNA viruses. All right. Good job, guys. Which of the following is not a type of viral capsid shape? Simple. Yes, that is correct. You all got that one. Awesome. Nice thing about having 5 players is you are all going to be on the leaderboard, which is Awesome. Which type of herpes virus causes chickenpox and shingles? Right, varicella virus. Yep, that is correct. Yeah, good to know the different kinds of herpes viruses. There are a couple of them and they all have different numbers. So study your herpes viruses and the difference between them. All right, next question. Which of the following is a reason the influenza virus can mutate so rapidly? Think about what makes it different from some other viruses. Yes, it has a segmented genome. Awesome job, guys. All right. Which of the following is the largest family of viruses? Largest virus family? It is Poxviridae, yes. Herpes viridae, not quite Poxviridae. Pox viruses are the biggest kind of virus family. And even though chickenpox is called chickenpox, it's not actually a pox virus. It's in the herpes family, but smallpox is in the Poxviridae family. Which of the following families of viruses has a disease which has been eradicated? Well, this one should be easy. I kind of just gave you the answer. Awesome job. It is Poxviridae, smallpox eradicated, which is fantastic. Which of the following families of viruses is a positive sense RNA icosahedral naked virus whose name means very small? It's a mouthful, but there will be questions like that on the exam. Yes, very good. Oh, Madison, you got kicked. Are you able to re-enter? Do you see the game pin still? Is it on my screen? All right. True or false? Antigenic drift is defined as small point mutations affecting the H and N constantly. Antigenic drift. Yes, that is true. Antigenic shift is the big one that causes pandemics. Antigenic drift is small point mutations. True or false? Viruses are cells. It just keeps loading. Oh, I'm sorry. Yep, they are not cells. Madison, I would recommend playing along and keeping track of the ones you get right. I'm very sorry. Kahoot is not the best sometimes. Which of the following is the third step of viral replication? Uncoding, yes. That is the third step. And attachment is the first step, penetration is the second, and uncoding is the third. All right. What strain of influenza virus caused the 2009 swine flu pandemic? It is H1N1. It is H1N1 was the swine flu. H2N3, I'm actually not sure what that one is, but H5N1 is the avian influenza. That's scary. But H1N1 is the. All right, next question. True or false? Prions are viruses. False, yes. Prions are not viruses, they're just proteins. They don't have genetic material. They're just misfolded proteins that are infectious. They're proteinaceous infectious particles. They are not viruses. All right, next question. What is an oncogenic virus? Very large virus, virus that only infects plants, virus that promotes tumor formation, or a virus that only affects the bones? It is a virus that promotes tumor formation. Awesome. We have 5 players. Were you able to get back in? All right. Which of the following is the best definition for the diploid cell line method of culturing animal viruses? So diploid cell line method. Yes, they in the diploid cell line method, they're derived from human embryos. They multiply for about 50 to 100 generations, and then they die. They're not immortal cell lines, um, and they can last for a couple of generations before dying off. True or false? Prions can be denatured by adding heat. False. Yes. So even though they are proteins, they cannot be denatured by adding heat like a normal protein. Think back to the list of microbes that are hardest to kill to microbes that are easiest to kill. Prions is at the top of that list. That's the hardest to kill. They can't be denatured by adding heat. They're very, very hard to destroy. Next. True or false? Prions replicate through transfer of genetic material. That is false. Very good. Yep, they're just proteins. They have no genetic material. They're really strange. All right. Which of the following is the best definition for pathogenesis? Very, very good. Awesome. That red one is just pathogen. The blue is disease. The orange or yellow, I've never known what color it is infection. But yes, pathogenesis is the way disease develops and all of its structural and functional changes. Awesome job. All right, which of the following is an example of a non-communicable infectious disease? Make sure you read the question here. Tetanus. So yes, this is a case of make sure you read the question. So a non-communicable infectious disease. You can't get cancer from someone else. You can get a cancer-causing virus like HPV from someone else, but you can't get cancer from someone else. Tetanus is an infectious disease. You cannot get it from another person. But here, no, let me rephrase everything I just said. Cancer is not communicable. It's not infectious. Tetanus is an infectious disease, but you can't get it from another person. Yes, sorry, I worded that in the worst possible way. All right, true or false? Transient flora describes microbes that come and go for a few hours to a few months. It is true. Yep, they're not resident flora. Resident flora is the one that like just stays in your body always. Transient flora, they come and go. Kind of like hotel residents, like the resident flora stay there always, and transient flora come and go. Which of the following is the best definition of endemic? Not epidemic, endemic. Yes, a disease that is always found in the region. That is an endemic disease. A disease that many people acquire in a very short amount of time, that's an outbreak. A disease that occurs occasionally in a population, or sorry, a disease that many people acquire in a very short amount of time, that's an epidemic. A disease that occurs occasionally within a population, just occasional outbreaks. But a disease that is always found in a region, that is endemic. Okay, who was Mary Malone? This was an extra credit question for us last semester. I don't know if it's one this year, but. Famous carrier of typhoid fever. So Typhoid Mary, she was the famous carrier of typhoid fever. It's a very sad story. She just wanted to keep cooking, but people didn't really know how to wash their hands back in the day. And so she just kept giving typhoid to people, and it was very, very bad. And they eventually had to lock her up. Sad. But yeah. Oh, I'm so sorry you got kicked out again, Madison. I saw that you made a second Madison. If you want to try one more time, feel free. It might be the Wi-Fi, but I'm so sorry that the technology is not quite being great right now. Which of the following is an example of direct contact transmission? Shaking hands. Yep. Not sharing from a drinking glass. That would be like indirect contact. That's indirect contact transmission. Shaking hands would be direct contact transmission. Okay. Which of the following is not an example of vehicle transmission? Yes, respiratory droplets. Respiratory droplets are carried in the air sometimes, but there's a difference. Like the line between respiratory droplets and airborne is kind of a fine line, but airborne is essentially if it can stay in the air for a very, very long time. Droplets are if you like cough it onto another person into the air, it's in the air for a little bit, but droplets are usually heavier and they can land on surfaces. Airborne is it can stay in the air for a very, very long extended period of time. So it becomes vehicle transmission when it's airborne, but it's indirect contact when it's respiratory droplets. Okay. A disease contracted from a mosquito bite is an example of what kind of transmission? Yes, biological vector transmission. Mechanical vector transmission is also a type of vector transmission, but that is when, say, a fly lands on, and this is gross, but it lands on like a pile of feces, and then it lands on your food. And the microorganisms from the feces land on your food, and then you eat it and get some kind of GI thing, probably. Biological vector transmission is when you are directly bitten by a vector. So in biological vector transmission, the microorganism is within the vector and replicates inside the vector. While mechanical transmission, it's just carried by the vector. It's not replicating inside of the bug itself. All right. Which of the following is a major contributing factor to the emergence of infectious diseases? There's a really good class called Emerging Infectious Diseases that I'm taking right now. That's a good book called Spillover. Yes, all of the above. So global warming is a contributing factor to the emergence of infectious diseases if you think about disease vectors specifically. So like mosquitoes, vectors, they like to live where it's warm. They survive while it's warm. So while the globe is warming up a little, more areas are staying warm for longer. That means the mosquito breeding season is longer, and that means that there are more mosquitoes carrying more infectious diseases. So all of the above are good examples of things that are impacting the emergence of infectious diseases. All right, true or false? Innate resistance is a non-specific defense mechanism against any pathogen. It is true. So yeah, your innate immune system, the one that you have when you're born, it's just very general. Basically, it recognizes anything that's non-self. It just kind of like starts attacking. Your specific immune system goes hand in hand with it, but that's the one that has a memory and is more specific. Okay, next. Which of the following is not a characteristic of innate immunity? Not a characteristic. Yep, awesome job, guys. Yeah, innate immunity doesn't have a memory. It just kind of attacks things that aren't you. So yeah, but it is activated by toll-like receptors. It doesn't have a memory, and it is present when you are born. Okay, next one. Which of the following is a characteristic of the second line of immune defense? Awesome job. Yep. A fever. So that's part of the second line of defense. Oh, did someone say something? Oh, no. So fever is part of the second line of defense. First line of defense is intact skin, flow of urine, mucus-coated hairs. Both the first line and second line of defense are part of your non-specific immune system, but fever is the second part of the non-specific immune system. So yeah, if things get past your like first line of defense, like intact skin, then second line of defense comes in with inflammation and fever. All right, which of the following is a granulocyte that makes up 60 to 70% of circulating white blood cells? These guys are important. Yep, and you guys recognize they're important too. That was fast answering. They are neutrophils, very important little cells. All right. True or false? T lymphocytes produce antibodies. It is false. So your B lymphocytes, some of your B lymphocytes are the ones that produce, become antibody producing cells. T lymphocytes, you have your cytotoxic T cells and you have your helper T cells. They're not antibody producing. They are part of the cell mediated immunity. B cells are part of humoral immunity and can produce antibodies. Okay, next one. What is the first cell to reach an area of injury? So say you get a cut. Oh no. It's all good. Just try your best. See what you remember from class. All right. Yes, neutrophils. They are the first cells to get there. If you get a cut through chemotaxis, cells kind of like just go to the area of injury. And neutrophils are the ones to get there first. They're like the first responders. Okay. What is the third step of phagocytosis? Cell eating. Yes, this is kind of a difficult question, especially if you haven't gotten to the steps. Definitely know your steps. I think the easiest way to memorize steps, I'm going to say steps another 5 times. A lot of the topics in these units are like mechanisms that have different steps to them. And the easiest way to remember those and their order is to come up with some kind of mnemonic phrase that works for you. So formation of a phagosome is the third step of phagocytosis. Chemotaxis is the first step, getting there. And yeah. Ingestion. Yes, chemotaxis. And so first it's getting there, chemotaxis is getting there, and then ingestion is the second step technically, and then formation of the phagosome is the third step. So ingestion does come first, but chemotaxis and ingestion are 2 separate steps somehow. They're similar. It's kind of like the difference between attachment and penetration of a virus. Oh, sorry. What's an opsonin? A virulence factor that some pathogens carry on their capsules, protein that inhibits phagocytosis, phagocytic cell, or a protein that promotes phagocytosis. Yes, it is a protein that promotes phagocytosis. Sorry. So adherence is part of viral replication. Adherence isn't really a part of phagocytosis. It like gets there, chemotaxis, and then ingestion, like enveloping the microorganism. But adherence is a part of viral replication. Yes, but an opsin is a protein that promotes phagocytosis. They're like the little, does anybody know what the specific antibody that is an opsinin is? Yes, opsinins do help with attachment. They help with, so yeah, I guess that's where it gets tricky. A lot of the words are used the same. In normal phagocytosis, there's not really an attachment factor, but opsinins can attach to the microorganism to make the ingestion step easier, I guess. And yes, it is IgG. IgG is the antibody that is used for opsonization. Yes, that is called opsonization. All right, next question. True or false? True or false? Interferons are proteins produced by some animal cells that interfere with the replication of bacteria. Oh, we're split down the middle here. Okay, so it is true that interferons are proteins produced by some animal cells that interfere with replication of microorganisms, but they are specifically, they interfere with viral replication. So we produce them, they're naturally occurring, and they inhibit viral replication specifically. They do nothing against bacteria. So this is kind of a trick question. And there are a couple of these on her exam sometimes where half of it will be true, but then part of it will be false. And so making the whole thing false. So make sure you're reading the question all the way through. Interferons work against viruses, but they do not work against bacteria. Okay, true or false? T cells originate in the bone marrow. Also split down the middle. So T cells and B cells both originate in the bone marrow. The way I think about it kind of is, yeah, and then the T cells go to the thymus to mature. The way I think about it is kind of like they're both born in the bone marrow, but then B cells live there, like they mature in the bone marrow as well. It's like B cells go to college, they are born in the bone marrow and they go to college in the bone marrow. T cells are born in the bone marrow and then they go to college out of state in the thymus. So yeah, and think T cell thymus, like B cell bone, kind of like B bone, T thymus, for where they mature, but they do both originate in the bone marrow, but they mature in different places. All right, all B cells become plasma cells, become memory cells, create antibodies, or mature in the bone marrow. Yep, it's mature in the bone marrow. So not all of them create antibodies. Some of them become plasma cells, some of them become memory cells, some of them create antibodies, but all of them mature in the bone marrow. Kind of another tricky one. All right. What kind of antibody class assists in opsonization? Oh, I asked this one earlier. This one should be another easy one. Yep, IgG. They're the little Ys that grab onto a microbe and help a macrophage eat it. All right, true or false? If you block attachment, you block infection. Yep, that's true. If a bacteria or virus can't attach to something, it can't infect you because attachment is the first step. So, well, in viral replication at least. So for viruses, especially, if you find a way to make them so they can't attach to a cell, they need a cell to replicate. And so if they can't attach, then you won't get infected. Okay. What kind of cell is the most active in cell-mediated immunity? It is the T cell, yes. So again, B cells are part of the humoral immunity. They float around in the blood and the lymph and deal with things that are extracellular. T cells are part of the cell-mediated immunity, and they fight off things that have already gotten into the cells, which is very important. Or they kill the cells that are infected already. Oh, and Zoom says we're running out of time. All right. What type of cells have CD4 plus molecule on the surface and assist B cells and T cells to react? Yeah, we have about 10 minutes of this Zoom left, but we'll try to finish up this Kahoot in that time. We can do 13 questions in 10 minutes. And then we'll move on to the next one. Yes, it is helper T cells. So what type of cells have CD4 plus molecule on its surface and assist B cells and T cells? So they assist. Helper T cells are helper T cells. They assist the B cells and T cells. They're your control center. They're in charge. Cytotoxic T cells are another kind of T cell. They're your killer T cells. But the helper T cells kind of have to tell those cytotoxic T cells, these are the ones you have to go kill. The cytotoxic T cells are your fighters, and the helper T cells are your ones that are just in charge of everything. And then cytotoxic B cells are not a thing. Yeah, your two types of B cells, they separate into plasma cells and memory cells, and your T cells separate into helper T cells and cytotoxic T cells. Okay. Which of the following is the best definition of humoral immunity? I keep going over the answers to questions before we get to them. Part of the immune system that uses B cells to fight off microorganisms outside of the cells. So remember, humoral immunity, extracellular, it's outside of the cells. T cells do the cell-mediated immunity. So glad none of you picked A or red triangle because it's not T cells that are involved. B cells are involved in humoral immunity. So you both got that, or you all got that bit. But yes, humoral immunity is not within cells. It is extracellular surface outside of cells. The extracellular fluid outside of cells. Okay. Which of the following cells does not have the ability to become a memory cell? It is plasma B cells. So yeah, you have your memory B cells, and then both helper and cytotoxic T cells can become memory cells, but your plasma B cells, they're just the antibody producing ones. Which antibody is the first produced in a primary immune response? So first time you get an infection, what type of antibodies produced first and the most? It is IgM. So IgM is very important in like telling when you were infected with something. If they do a blood test to see what you've had, if there's a lot of IgM antibodies, they're like, oh, you just got this infection for the first time. You haven't had it before. So it's very important in diagnosis of disease. IgM is produced the most and first in a primary immune response. IgG is produced the most in a secondary immune response. So like your second infection with something. So there's a lot of IgM like after you get a vaccine or after you get your like first infection with something, and then IgG, your second encounter with things. Both are produced in both responses, but like more is produced, more IgM in the first one. Which of the following is the best definition of artificially acquired passive immunity? So artificially acquired passive immunity. Yep. So immunity acquired after injection of preformed antibodies. So it's passive, so you're not making the antibodies yourself. In active immunity, your body is making the antibodies. And if it was naturally acquired passive immunity, blue would be correct. But artificially acquired is an injection. Okay, next one. What is the best definition of epitope? It is a specific portion of an antigen that an antibody recognizes and binds to. It's not the part of the antibody that binds and recognizes a part of an antigen. There is a good picture in her slides that I will show in the next portion of the Zoom that defines epitope pretty well because it gets a little bit confusing. All right. Which of the following is not an example of a whole agent vaccine? So vaccination PowerPoint was one that you guys were responsible for going over on your own. So we have some vaccine questions. Very good. So yeah, live attenuated and killed vaccines are both examples of whole agent vaccines, like it's the whole thing, the whole microbe. Conjugated vaccine is a part of it. Next one. True or false? Live attenuated vaccines are perfectly safe to give to immunocompromised individuals. Yep, that is false. That's why we need to vaccinate everyone around them so that it can't spread and get to immunocompromised individuals. But because they're live attenuated vaccines, or it's a live version of the microorganism, and there's a risk of it transforming back into its like infectious form. All right, what kind of vaccine is the seasonal injected influenza vaccine? All right, we're perfectly split through these ones. So yeah, look at the vaccination PowerPoint, guys. So it's the flu shot is an inactivated virus, so it's killed. It is a whole agent vaccine, and the injected version specifically is inactivated. I believe the nasal spray is live attenuated, but it is the whole flu virus in both, so not conjugated or subunit. All right, next one. What kind of vaccine is the MMR vaccine? MMR is live attenuated. So yeah, definitely look over those. You should know examples of each kind of vaccine. All right, we're down to the wire. We have a minute and a half or 2.5 minutes to get through 3 questions. True or false? Conjugated vaccines are a type of subunit vaccine. It is true. Conjugated vaccines are a type of subunit vaccine. Not all subunit vaccines are conjugated vaccines, but conjugated vaccines are a type of subunit vaccines, kind of like a rectangle and a square. What is a toxoid vaccine? Yep, made from modified inactivated bacterial exotoxins. So if you guys remember learning about those, like Botox, the tetanus vaccine is a toxoid vaccine, and viruses don't make endo or exotoxins. All right, true or false? mRNA vaccines change your cell's DNA. It is false. This was the whole big debate. It doesn't actually change your DNA because it's an mRNA piece of it. So the mRNA goes into your cytoplasm and forms a protein, but it doesn't change your like base cellular level DNA. And then last question, which pathogen can be prevented by a subunit vaccine? It is the Haemophilus influenza B, so that is a bacteria. Flu, again, is live attenuated, or well, the nasal spray, or killed for the injection. Rabies is, I'm blanking, but it's not a subunit vaccine. It is a whole vaccine as well. And then rubella is part of the MMR vaccine, which is a live virus vaccine. So Haemophilus influenza B. Study your vaccines, and then this Zoom is going to kick us out in like a minute. So same log-on information, and we'll go over any questions that you guys have. And I'm just going to end this now so I don't get cut off mid-sentence, but see you guys in the next one.